Hello and good evening everyone. Today I'm going to present my research titled Investigating the Relationship Between Behavior, Environment, Genetic Diversity, and Fitness in Queens of the Neotropical lands Camponotus renggeri and Camponotus rufipes. This research is being developed by me, Marianne, Henrique, Anete, Paulo, and Gustavo. I'd like to begin this presentation by defining fitness as the ability of an organism to pass on its genetic materials to its offspring. Research on this topic is of great interest for genetics and evolutionary biology, as it allows this information to be an important tool in understanding patterns of natural selection and helps us understand how various factors might shape reproductive success. Here, I'd like to separate some of these factors into two simplified categories. The first are endogenous factors, which are intrinsic features to the organism or the individual itself, such as its genotype or phenotype, its mating success, natural history, age, and much more. The second category I'd like to mention are exogenous factors, which mostly relate to environmental and habitat characteristics. However, this research deals with a specific group of animals, social insects, and more specifically, the ants. These insects are famous for their eusocial way of life, but this brings forth an extra layer of complexity to the scheme, as we also have to take into account the colony structure, which, among other things, can relate to mean colony relatedness, polyandry, and polygyny, which are, respectively, when a single queen mates with multiple males and when multiple queens contribute to the same colony. We know that fitness is driven by many different variables, but this begs the question, how do multiple factors influence individual queen fitness? In order to answer this question, we may begin speculating some of the expected relationship between these factors and fitness itself. Starting with fitness and polygyny, we know that queens, for the most part, are the sole responsibles for the reproductive function inside a colony, meaning that every worker, males, and possible future queens are her offspring. However, when another queen is part of the same colony, reproductive partitioning might be at play, meaning that both queens share this function, reducing their reproductive success. Beyond that, it is known that queens of polygynous colonies tend to be less fertile and live shorter lives, reducing their overall fitness. That being said, it is not unreasonable to expect a negative relationship between polygyny and fitness. As for polyandry, this strategy is one of the drivers for genetic diversity within colonies, but it comes with costs to the queen, such as being predated, injured, or infected by a disease. On the other hand, queens that mate multiply have a larger sperm storage than those who don't, which would guarantee a high fertilization rate to produce a large number of workers during the colony's lifespan. Therefore, we can assume that polyandry and fitness might have a positive relationship. Moving on to parent heterozygosity, in this case specifically queen heterozygosity, things get a little trickier. The overall relationship between heterozygosity and fitness is highly debated, as it involves many different variables, such as the relevance of genome-wide heterozygosity versus heterozygosity on functional loci. It is known, however, that heterozygosity has some relationship to inbreeding detection and might be relevant to avoid negative effects related to inbreeding, such as inbreeding depression or passing on deleterious alleles. So, heterozygosity might have a positive effect after all. For the last factor, I'd like to consider habitat quality. Colony foundation is one of the most important steps in the life of a young queen, and while it might not have a direct effect on its ability to produce offspring, a stable and safe environment guarantees less stress for the queen, as well as more resources around the nest for the colony to thrive. Also, it has been shown that ant colonies in disturbed areas might be smaller and invest less in maintenance, which means that workers are produced less frequently. Therefore, we may conclude that a high-quality environment might benefit queen fitness. With this simple train of thought, we have now established 4 hypotheses that describe what might drive individual queen fitness. To test this hypothesis, we chose to look at 2 closely related ant species that are highly abundant in the Cerrado, Camponotus renggeri and Camponotus rufipes. Despite both of these species displaying interactions with many plants and other arthropods, they have contrasting nesting habits and breeding strategies. Camponotus renggeri tends to nest mostly on dead tree trunks and has a lower mating frequency, whereas Camponotus rufipes may nest using dry straw, dead trunks, and even underground, while presenting higher mating frequencies. The taxonomic proximity, ecological differences, and abundance of the species make them interesting models to study what may affect queen fitness. In order to conduct this research, from 9 to 20 workers were sampled from each colony and genotyped. Then, lineages were reconstructed, resulting in 40 queens for Camponotus renggeri from 11 polygynous colonies and 44 queens from 17 polygynous colonies for Camponotus rufipes. With the reconstructed genotypes, we were able to determine the number of queens per colony and the number of male mates per queen. As for fitness measurements, we used reproductive success as a proxy and calculated the proportion of offspring per queen in each colony. For polygyny measurements, we used the obtained number of queens per nest, and for polyandry, the number of male mates per queen. Both measurements were obtained using the Colony 2.0 software. Using the genhat function in R, we calculated the proportion of heterozygous loci per queen. For habitat quality, 10-meter buffers were plotted around each nest, which correspond to these species' home ranges, and the proportion of Cerrado vegetation was calculated for each of them using the landscape metrics package in R. After that, correlation tests were conducted between each of these variables and fitness in order to obtain our answer, all of which had a confidence interval of 95%. Moving on to our results, we obtained that Camponotus renggeri fitness has correlations to polygyny, queen heterozygosity, and habitat quality. This may be explained by the fact that these species present less diverse nesting habits, thus, the surrounding environment quality might be more relevant to their success. Beyond that, co-occurring queens are less related, meaning that reproductive partitioning might have a strong effect on them, which might explain the strong negative correlation between fitness and polygyny, as we expected. Also, the species' low mating frequency might be the reason why polyandry was not a statistically relevant correlation. As for Camponotus rufipes, all correlations except polygyny and fitness were not statistically relevant. This result does not match our expectations and might indicate that there are other drivers to queen fitness that were not used in this research. It is interesting to notice that the relationship between polygyny and fitness in this species, although negative, is weaker than in Camponotus renggeri. In fact, co-occurring queens of Camponotus rufipes tend to be more closely related to one another, so reproductive partitioning might not be as relevant here. Also, the more diverse nesting habits that these species display might be one of the reasons why the environment was not a significant factor in this case, indicating that these ants are not as dependent on external factors. As for the high mating frequency, it may only serve as a driver of intracolonial genetic diversity and may not be of much relevance to individual queen fitness. Beyond these results, it is important to notice that the driving factor used for this analysis are not isolated in the real world, and represent only a simplification of the complete system. In fact, polygyny and polyandry are known to be negatively related, and it is highly likely that other correlations exist between these variables. Beyond that, we must not forget that correlation does not imply causation, but it might sometimes be a good indicator of possible relationships, seeing that causation does imply correlation. In order to address these shortcomings, we will continue developing this research by using a method that allows us to explore causal relationships, such as structural equation modeling, hereby represented by this summarized model that is being currently developed by our team. This method allows us to establish a clear path of causality, in which each factor might have direct or indirect effects on the rest of the system. Also, we are now able to separate this system into different scales of magnitude, with gray being related to colony-level factors and white relating to factors that affect individual queens. I'd like to thank the workshop scientific committee for the opportunity to share our findings, and FAPESP, CNPq, and CAPES for financially supporting our work. Also, thank you all for your attention, and I'd be happy to answer any questions that you might have.